How?
Hi, Fed, would you like to welcome everyone to the Week 4 MOOC Hangout Q&A session, please?
Yes. Hello. Good afternoon, everybody. I'm very happy to welcome you on this session. This is the fourth week of the MOOC in vaccinology, and you know that we have every week on Mondays or Tuesday we have a Hangout live session in which you can either participate or you can attend. Just look at what is said during this session. It's a choice. It's a moment of choice to ask your preferred question, because you can have in these sessions the lecturers that you've seen the videos online, and you can ask them some precise question that you not been answered in the chat on the MOOC. So this is the occasion, and I would like to remind that it's a good opportunity to participate, and also if you can't participate, you can ask by chatting on the discussion forum. You can ask your questions, then GB will collect these questions, and we can answer them in live during this short session. It's a short session, only half an hour, so we have to be fast. So thanks, GB, to recall us the questions that have been raised for this session. Thank you.
Okay. Oh, thank you. Today we have Dr. Engelhardt, the flu vaccine expert from our MOOC Vaccinology. Today I collect a question from the discussion board, and there's a question on the developing of DNA vaccine for influenza, and the student would like to know that the promising of DNA vaccine for influenza, and how would we tackle the possible interference of exogenic DNA with our endogenic DNA. And if you can give some other reference for the reading, they would really appreciate it.
Okay. All right. Well, thank you for the question. I think if you're talking about DNA vaccines for influenza virus, you have to separate out two questions. One is to address the question of vaccines against flu in general, and there are many concepts that one could take. And the other is the question of delivery, and that would be the DNA vaccine part. And there are, in different regulatory systems, there are different regulations and guidelines and guidances on how new biotech products need to be developed, what needs to be taken into consideration, and how they will be evaluated. And so if somebody's thinking about a DNA vaccine, they need to take note of these guidelines and develop the product accordingly. The important question with relating to flu is, what is your concept? How are you going to address the question of inducing immunity against influenza virus? Are you going to make this an annual vaccine? Are you going to make this a broader vaccine that maybe doesn't need to be given annually? So I think these two questions are sort of, they are of course related, but also to a certain degree separated. And I really cannot say anything about the promise of a DNA vaccine. I'm not an expert on DNA vaccines. I'm not sure there's a lot of activity on DNA vaccines in influenza at the moment. There may be some, but I'm not really aware of it. Yeah, I guess that's where I can stop at the moment.
Okay, thank you.
Maybe, GB, I can add a word on the general on DNA vaccines.
Okay.
Yeah,
yes, please.
Yes, just to say, so thanks, Otmar. I forgot to say that Otmar is a researcher at the NIBSC Institute in London. Thank you for participating. So DNA vaccines, yes, DNA vaccines are a very promising strategy. However, up to now, this strategy is not able to work properly in human trials. Many DNA vaccines have been tested in human trials, and apparently what is missing is a way of triggering enough innate immunity against this DNA. And the question in this regard was quite good, because probably this is because we are not able to make the distinction between our DNA and foreign DNA. So DNA is not enough able to stimulate immunity in humans, although it is able to do so in mice if you add some very strong adjuvants or if you make electroporation of DNA, which is very difficult to use in human in a great scale, in a large scale. However, this is still on work, and ongoing assays are being done to try to discover new adjuvants that might be able to help DNA to induce the good immune responses in humans. But this is for sure not ready for now.
I think you're muted, Chibi.
Yeah, you mute, Chibi. We don't hear you.
And that's the DNA vaccine for the flu. And also another question for the flu vaccine. They also interested about the universal influenza vaccine. So is that it will make it like we don't have to create the new vaccine every year. But is there any progress on the flu universal vaccine?
Okay, I think this is a very pertinent question, a very topical question. There's a lot of activity ongoing at the moment in the field of universal influenza vaccine research. There are a number of concepts that people are pursuing in this. Some of them, such as conserved epitopes in the hemagglutinin protein, the stem. There are conserved proteins in the influenza virus, such as the extracellular domain of the M2 protein, the so-called M2E. And then there are people who are, for instance, working with peptide vaccines, trying to find conserved peptides that they can use to get a broader immune response. Most of these concepts at the moment are still at a very early developmental stage, many only in animal models. There are some concepts that have been taken into clinical trials, phase 1, perhaps phase 2, but I don't think much more than that. But it's a very active area of research where certainly in the next few years things are going to happen. And for instance, I know that in the United States there's a big push towards universal flu vaccine, and I understand there will be funding available for universal flu vaccine concepts. So I would say watch this space. There are certainly developments, but as in many cases, they need to be tested in humans, and the proof will be whether in clinical trials they can prove to be more broadly reactive. In fact, the regulatory questions, I think, are quite serious in this case. How are you going to prove in clinical trials that your vaccine is going to be universal? I assume that you can first show in a clinical trial that your vaccine may be more broadly reactive, but universality will be a big challenge and will probably only happen over time. Thanks.
Great. Thank you so much for the answer. Right now we have Dr. Stenger, Stefan Stenger joining us. Hello? Hello? Hi.
In Germany. Can you hear me?
Yes, we can hear you.
Hello, Stefan.
Hi.
How are you doing?
Hi.
Hi.
We cannot really see you quite well. And I actually have Dr. Hart on the phone. She's having a problem connecting through her computer, but now she's currently on the phone with me. Hello? Hello? Hi? Yes. So we're going to do some question answer with her first. Hopefully it... Oh, I just lost her. Anyway, I'm trying. But
this is a difficult job. Thank you, GB.
So maybe we'll ask question to Dr. Stefan first. Where is the question now? No, I'm lost. Somebody was suggesting to me too that I should have someone with me to take care of the connection while I'm like moderating the video session. So maybe I'll ask the different question for now first. There's a question on the pertussis vaccine. Maybe Frederick, if you could help answer it.
Yeah.
They're mentioning that because in the session they said in the class said that pertussis is only a pediatric disease, but is that yes or no? And if the adult who are already vaccinated with pertussis, what would be the outcome if the person get another vaccine of the pertussis?
Yeah, so there are two questions on that. The first question was, is pertussis a pediatric disease? Of course it is, but it is not only a pediatric disease, because these days we know that even after vaccination, there are a number of population that are at risk to get pertussis, whooping cough, and especially pregnant women and elderly. So these individuals are also at risk of pertussis. So for this reason, pertussis is not only a pediatric disease. It is a pediatric disease, but not only a pediatric disease. So now, is there any problem to get vaccinated against pertussis, even if you got this vaccination when you were an infant? No, there is no problem, because you will be boosted by the vaccine, and currently it is recommended that you should be vaccinated against pertussis every 10 or 15 years. So this is a normal way of immunization against pertussis. This is my answer.
Thank you. I've been getting a call from. Whew, okay, I think because Corinne trying to connect to the session, I feel so bad that I cannot help her out with this, because because we have some question on the HPV vaccine as well, and I really want her to answer the question, but it's been another technical problem once again. Okay, for Dr. Stefan, hi, thank you for joining us. The question on the BCG vaccine that I collect from the discussion board, people were wondering that the new vaccine strategy, is there any product come out from the new vaccine strategy for BCG, and would it affect with the skin test? Because like the BCG, if the person get the BCG vaccine when they were young, if they get the skin test, they do have some reaction from that. So is there any new vaccines on the BCG that come from the new strategy that you present in the class? And is there any reaction with the skin test? And how can we distinguish the person from getting the person already vaccinated or the person who get infected by TB? Thank you.
Yes, so I'll be happy to answer that, to take that question. So first of all, there are several examples for far advanced vaccines. One big group of the vaccines is based on molecularly improved BCG strains. They're very far advanced. They carry additional antigens or additional mutations, but the disadvantage remains. All of these BCG-based vaccines will cross-react with the skin test. So even with the modern and new vaccines that are being developed, all of which are based on BCG, will cross-react with the skin test. A second major strategy where much success has been achieved is to use vaccines which are not based on BCG, but which use defined mycobacterial antigens together with an adjuvant to provide efficient protection against tuberculosis. And some of these vaccines are, or some of these protein subcomponent vaccines, which they are called, contain antigens which do not cross-react with BCG. For example, ESAT6 or TB10.8 would be examples. And if you use these proteins for vaccination, which has been successfully done in animal models, you will have no cross-reactivity with the skin testing, or even not the other immunological assays. So even though the majority of the new vaccines are BCG-based and will cross-react with skin testing, there are advances in terms of creating vaccines which are not BCG based and which will not cross-react with skin testing. Thank you.
Thank you so much. So for the rest of the question is on the HPV. We tried. I'm not sure. Maybe
there were some questions on the RSV too, also.
Oh,
AD.
Yes.
Maybe either Otmar or I can answer these.
Okay.
Depending. Can you re-ask them, please?
Oh, yes. There's a question on RSV. Does antibody enhancement impact? Is there any antibody enhancement impact from the RSV? Is it occur naturally in the RSV infection? Is this mechanism similar to dengue infection? So that's the question on the RSV. Yep.
Otmar, you want to answer that, or?
I can start, and I think I'll then hand over to you. So I'm really not an expert in RSV, although I'm starting to get involved a little bit in the RSV field. So the antibody enhancement of disease, which was seen with formalin inactivated vaccine in early trials, was a real setback for the RSV vaccine field, and has hampered the field for decades. But now there are different strategies being explored, and I think the basic idea is that you would use probably live attenuated vaccines for children who are naive, in order so that you could avoid any such potential antibody enhancement of disease, and that subunit vaccines or other types of dead-killed vaccines would be used in non-naive subjects, such as in pregnant women for maternal immunization. As to the mechanism, I'm really not an expert on this, and I guess I won't try to answer. Maybe I'll hand over to Frederick at this point.
Yes, the antibody enhancement is due to the action of antibodies that are not enough neutralizing, but sufficiently able to bind the virus, then to bind to other cells that express Fc gamma receptors that are able to bind to this part of the antibody. So this antibody, in fact, will enhance the infection of a number of cell types. And this is, in fact, this is not only theoretical. This has been observed in the primate model. This has also been demonstrated using sera from children with RSV disease that has been used in vitro to show that when you pre-incubate these antibodies with the virus, you can enhance the infection of different cells. It is a similar mechanism that occurs in dengue, and it has been shown mainly with vaccine, with former vaccine that you were talking about, the formalin, the former formalin inactivated RSV vaccine, that also been shown with a formalin inactivated measles vaccine in the 60s that also did exactly the same, showing more severe disease among children that have been vaccinated with this vaccine as compared to the the control group. The theory now is that if you elicit antibodies to a virus without simultaneously eliciting T cells, CD4 and CD8 T cells, you may have antibody-dependent enhancement of the disease. That's why, as Otmar was saying, for first immunization, you need some vaccine candidate and that contains that contain antigen able to elicit T cell response. And we know that inactivated vaccines are not very good at that. That's why, for example, for a flu vaccine, you just when you immunize adults with a flu vaccine, you just boost the former response that they already have, and it's because these individuals, they had a flu in your life, so they have T cells already in place against flu. So you can boost them with an inactivated vaccine. But if you use an inactivated vaccine directly in naive individuals, and this has also been recently shown in a recent phase 3 trial with a vaccine against dengue that was not able to induce enough strong T cells against dengue. That in the youngest population, in fact, the disease was enhanced in the vaccine group. Excuse me. Although I'm in the south.
I'm sorry.
Excuse me for a moment. I have to drink something.
Oh, okay. Would you need a break?
Yeah, 2 minutes.
Okay. So, I have a question on the HPV. I'm not sure if maybe any of you, sorry, if any of you could answer this, because there's some serious side effect that been on the internet anyway on the HPV. They were wondering, like, do we know the causes of the particular serious side effect that caused by HPV, and or this is just a person case-to-case basis? Fred is back.
Yes, I'm back.
Hi. Sorry. One moment. Okay. As you can see, I'm in the lab and everybody's very active today.
And as you can see, the boss is not in the lab.
Okay, yes. There's some question on the HPV vaccine because there's been some anti-vaccination movement, and they said that HPV have a serious side effect. Do we, as a scientist, do we know the cause of this particular side effect, or this is just a case-to-case basis? Is no link or no evidence for example?
Maybe Stefan, can you answer that?
Not really.
Not really.
To be honest, I don't know. I have no idea about the side effects of the HPV vaccine.
Okay.
Sorry.
Yeah.
Got one on BCG or tuberculosis, I'll be happy.
Oh.
but viruses.
So maybe we can talk about the serious side effect of vaccine in general. Is there any particular case of serious side effect of, for example, BCG vaccine?
Yeah, thanks for that one. So in BCG, as with many live vaccines, we have the problem that it's been to newborns immediately or several hours to a few days after birth. And here, especially in Asian countries and in some African countries, we have the risk of vaccinating infants which are born to HIV-positive mothers or which have a severe combined immunodeficiency syndrome, meaning that the attenuated BCG strain will, in these patients, HIV-positive or suffering from a severe immunodeficiency, cause BCG-itis. And that can be a local effect, which is painful and will scar, but even worse, it can cause disseminated BCG infection and even lead to the death of the infants. So this is one of the major arguments why we are looking for a vaccine which is not based on a live BCG strain, or alternatively look for BCG strains for vaccination which are much more attenuated than the parental BCG strain which is used at the moment.
Okay, thank you. For Atma, is there any serious side effect of the flu vaccine, and that you can know of?
Maybe I can just interfere on that one. if I may, because I just got my influenza shot yesterday, and my muscle is aching, I can tell you.
Yes, I had my flu shot last week and was fine.
Well taken, it's only minor side effect.
Yeah. I think generally current influenza vaccines are very well tolerated, and they are the usual local side effects that can occur, like swelling, pain at the injection site, and those things. And occasionally, sort of unexpected events have been seen, like the narcolepsy cases in Europe with the adjuvanted AS03 adjuvanted vaccine for the H1N1 pandemic virus. And that's obviously a bigger issue. And there seems to be some genetic element. Certain people with a certain HLA type seem to be predisposed, but only a very small percentage of these people actually go on to develop narcolepsy. And in the past, in 76, 77, there was an instance of Guillain-Barré syndrome, but that hasn't really been seen since with influenza vaccines. And that was for a particular, the so-called swine flu episode of 1976 in the US.
So
it's a pity that we can have Karin here, because she would have been the ideal person to answer these questions about the.
She would just call in me, and she will give me the answer in writing, and I will put it on the discussion board for the answer for the HPV. I think we had a very fun, successful session today. Thank you very much, and thank you, the IT Angel, that we got our instructor to join out. Fred, would you like to close the question?
Just a comment. It's a pity that it's too short, because I think it's interesting, and it would be interesting to know how many people, in fact, are following the discussion. It's important because when we discuss online like that, it's possible that a large audience is following us. So particularly on these questions of adverse events that are really tough questions regarding vaccines, we need the right answers to the public, of course, and to the students, because in this MOOC, the audience is broader than in a classroom, of course, and these answers are very important. So it's a pity that Karin has not been here, but it was fun. Yes, I agree. It's very interesting to talk like that, and it's a pleasure for me to have Otmar and Stefan together with us in these sessions.
To be honest, I had to use the computer of a student of mine, because the one in my office isn't equipped with a camera and with a microphone. It saved me. So it was a pleasure. It was really good, GB, and thanks a lot. You were very kind in preparing this. Thanks.
Thank you.
Yeah, thanks a lot.
Thank you. It was really good.
Okay.
Okay. We see each other in March for the course.
Yes, we will.
At Paris, in Paris.
Okay. Okay.
Thank you very much, GB, for this.
Thank you, everyone, and I think we'll probably see everyone soon. Okay. Thank you.
Okay.
Bye.
Thank you. Bye.
Bye, everyone.
Bye.